Good morning, students. In our previous lectures, we studied about different classification of angiosperms given by several authors, including modern classification, then primitive angiosperms, origin of angiosperms, phylogeny of angiosperms, and many more. Now, coming to the practical aspect of the taxonomy, today we will discuss plant collection and specimen preparation. Plant collection and field preparation of specimens are the fundamental aspects of study, training and research in plant systematics. Herbarium specimens are the permanent record of plant species of a particular place at a given time. Therefore, the plant should be carefully selected, and the herbarium specimens should be properly prepared and preserved. So, which type of specimen should be collected? The points to be kept in mind during a plant collection are: collect entire, vigorously growing, typical specimens. Select such individuals that represent almost all phases of natural population. Then avoid collecting insect damaged specimens. Collect underground parts, that is root, bulb, rhizome, tuber, etc. of herbaceous perennials. Collect those specimens of flowering plants that contain flower, fruit and seeds because keys are prepared mainly on the basis of these characters, floral characters. Specimens larger than the size of a single sheet should be divided and pressed on a series of sheets. This we will see when we are pressing the specimen. Collect plants with the leaves intact, as different kinds of foliage prove helpful in identification. Then collect the bark and wood sample of the woody plants because they are also key characters when we identify a plant. Avoid collecting rare or uncommon plants. Never collect the only plant of a species at a locality because it will going to be damage biodiversity. So never collect the only plant of a species at a locality. We should wait at more plants of the same species grow, then we go and collect the same plant. The next step is while collecting the plants, field equipment and supplies. Some of the commonly used field equipment and supplies needed for plant collection and preparation of herbarium specimens are field press. It is made up of pair of hardwood, metal or strong plywood frames of 12 by 18 inches. That is field press. Then dryers and blotters. These are of sheets of heavy blotting papers or the other moisture absorbing material of 11 by 16 inch dimension. Old newspapers are also used as dryers. Sorry for the interruption. Now comes straps or ropes. Rope such as cord or a pair of strong web straps of about 4 to 5 feet length are used to tighten the press. These are required for tighten the press. Then corrugate ventilators. Sheets of corrugate cardboard of 12 by 18 inches are used in between the dryers or plotters as ventilators when plants are dried by artificial heat. They provide space for the passage of air through the press to remove moisture. So this is the function of corrugate ventilators. Now field notebook, very important part, field notebook. An indispensable item of a plant collector is a permanently bound small field notebook which with ruled pages. It is used to record the full data such as date, place, locality, habitat, elevation, local name, collector number, etc. of the collected plant. The data is recorded in the field notebook in the field itself. Now, digging and clipping tools. Trowel, diggers, hammers, pruning shears, garden clippers, geological pick, and heavy sheath knife, etc. are all used for digging and clipping of the. We can see here the tools. This is digger. Now, string tags, these are made up of waterproof material and are used for labeling plants that are not immediately pressed. Now, vasculum. Vasculum is a container made up of tin or aluminum sheet and contains a hinged light lid. Plants which are not pressed in the field are placed in a vasculum to preserve their freshness for some time. This is vasculum. This is vasculum. Collecting bags. These are plastic bags used as containers for fresh specimens. Collecting bottles. These are glass or plastic bottles with leak-proof screw caps. These are used for collecting small-sized material to be preserved in liquid preservative. Now, liquid preservative, formalin, acetic acid, alcohol. FAA is the most common liquid preservative used for anatomy materials. 6 is to 3 is to 1 mixture of chloroform, 95% alcohol and glacial acetic is often used for cytological materials. So these two liquid preservatives we use. Next comes to hand lens. A 4x or 10x, 5x or 10x lens is used in the field for observation and identification. Waxed paper. Sheet of waxed paper are used for pressing viscid or weak looking plants. Cardboard storage box. These are boxes of containers used to store dry material. These are cardboard storage box. Then maps. These are useful in the field for determining the locality of the particular species. Maps. Then color charts. Color charts. These are used for determining the actual color of the flower part in the field itself. This is the use of color chart we have with us when we go for collection. Camera and film. These are used for taking photographs of important plants in the field. Very important part. And nowadays we have mobile, so nothing to worry about it. And other field equipment and supplies, altimeter, compass, pocket knife, soft lead pencils, insect repellent, portable plant dryer, and seed envelopes are some other useful equipments for plant collection. So this is all about the field equipments which are very important. We see this is plant press, this is vasculum, this is digger, and these are cutters, all cutters, and this is herbarium. Now, how we prepare specimen. So first, organization of the field press. Second is pressing of a specimen. It is very important how to press the specimen. This will decide the specimen will be valuable for us or not. So, specimen after being cut or dug should be pressed as soon as possible. This specimen can be placed carefully on a pressing sheet, that is a newsprint sheet or a blotter, taking care so that there is no folding or overlapping of parts. The extra leaves or branches are removed if necessary. Plant too large to fit in 11 into 16 inches fold of a newspaper or a blotter may be bent into V, N or M or W. The specimen should not protrude from the fold of the paper. Specimen should be arranged in such a way so that some upper and lower surfaces of the leaves are exposed. Flowers or inflorescence should be spread out thoroughly for a complete view. A few flowers can be cut longitudinally and pressed if possible. Each sheet should contain a collector's number, which refers to the notes in the collector's field notebook. We can easily find the uh, notes which uh, taken in the field while collecting the same plant. In the press, the specimen in this specimens in the specimen paper are placed in between two dryers or blotters. A corrugate ventilator is often inserted before the next specimen paper and dryers are added. All specimens are placed for pressing in such a way so that the entire bundle is ultimately of almost uniform thickening at the middle and sideways. The press is now ready for drying. Now comes drying of the specimen. A specimen should be dried as rapidly as possible so get the best results. The usual process of drying, the press containing the specimen is placed in the sun. After about 24 hours, the press is opened and the specimens are placed in fresh waters. Any last arrangement of the plant part may be made at the same time. The press along with the specimen in fresh blotters is now again bound tightly. The wet blotters are dried in the sun for reuse. For 3-4 days, the wet blotters and dryers are changed daily until the specimens are completely dry. Artificial heat may also be used for the drying process. Now comes mounting of the specimen. The process by which a dry specimen is attached to a mounting paper or a herbarium sheet and the label is affixed at the lower right corner of the sheet is called mounting. The standard size of a herbarium sheet is 28.75 by 41.25 centimeter. Heavy handmade card sheets are used as herbarium sheets. Before mounting, the dry specimens are dipped in a saturated solution of mercury chloride and methyl alcohol to prevent infection of fungi, surface sterilization, infection of fungi, insects, etc. In place of mercury chloride, lauryl pentachloronate, pentachlorophenate is also used because the former is a violent poison. Mercury chloride is poisonous. A good quality glue or paste is applied to the back of the specimen for affixing them onto the herbarium sheet. Common animal glue is used in India for this purpose. Use of cellulose tapes or stitching of the plant part with thread should be avoided. Now comes deep freezing methods. These days, deep freezing methods are being adapted in some herbaria to prevent infection of fungi, insects, etc. instead of mercury chloride and other harmful chemicals. So, in this, we use portable freezer unit and deep freeze aquatic wall. Portable freezer units offers two main benefits over traditional ice coolers. First, refrigeration control offers temperature stability independent of exterior temperature without the worries about the ice melting. Second, such units can functional when well in remote locations where replenishment of ice may not be feasible. And the deep freeze Arctic vault is recently in the early months of 2010, thousands of seeds, plants, and crop varieties from several countries belonging to all continents have been shipped and preserved in a vault in the Arctic Circle. According to the organizers of Arctic Circle, these would be preserved here in deep freezer for thousands of years for future use. Now comes labeling of the specimen. A label usually of 6.5 by 10.5 centimeter dimension is pasted on the lower right side of the herbarium sheets. It contains definite information about the specimen pasted on the herbarium sheet. Ideally, it contains a miniature easy on the plant and its habitat. The paper of the label should be of high red quality. Following information should be incorporated in a label. It is very important. Labeling of a specimen. Heading indicating the name of the institution or person along with the name of the state and country. Now, next is family name, scientific name, that is name of genus, species along with authority by whom the name is given. Locality indicating the place of collection with its latitude and longitude and distance from the well-known place. This is called referencing. Habitat indicating various vegetation types, moisture, soil, slope, etc. Date of collection, field note from the collector's field notebook, collector mentioning his her name, collection number, which is a numerical series starting with 1 and continuing throughout the lifetime of the collector. Local or vernacular name of the species and additional information if there is, such as color of the flower and leaves, associated plants, bark, economic uses, etc. And last comes the identification of the specimen. Finally, pressed, dried, and labeled specimens are identified with the help of appropriate florals, manuals, and monographs. A duplicate specimen is to be retained before sending a specimen of the specialist for sending a specimen to the specialist for identification. So, this is all about the plant collection and specimen preparation. I hope you have no doubts, you understand all these about plant collection and specimen preparation. Thank you.